The word blood evokes lots of thoughts, from tiny paper cuts to major injuries. Spilling that red liquid is almost never a good thing. That's because blood helps us move nutrients and waste around the body, as well as regulate our pH level and prevent infections. In fact, some components of blood help prevent the loss of blood during an injury. The components of blood can be separated out by simply spinning the blood in a centrifuge, a machine that whips a vial of blood in a circle over and over, really fast. This is kind of like what happens to clothes in a washing machine. When blood is centrifuged, the heaviest blood components move to the bottom, and the lightest ones move to the top. Overall, 3 distinct layers form: the erythrocytes, or red blood cells, at the bottom, the buffy coat, which contains platelets and immune cells, in the middle, and then the plasma on the top. So starting at the bottom of the tube, there's the large layer that makes up approximately 45% of the total blood volume, which again is made up of erythrocytes. This value is called the hematocrit. A decreased hematocrit means that there are too few erythrocytes, either because they're not being made or because they're being destroyed. On the other hand, an increased hematocrit can be due to dehydration, because if there's less liquid in the blood, then the portion taken up by the erythrocytes would rise. Alternatively, there might simply be too many erythrocytes being made, which can happen in some diseases. Now, the main function of erythrocytes is to carry oxygen to the tissues, as well as bring carbon dioxide to the lungs so it can be expired. Erythrocytes are shaped like thin biconcave discs, meaning they have a depressed center which makes them flexible enough to fit through even the smallest blood vessels. This shape also increases their surface area, which helps them conduct gas exchange efficiently. Erythrocytes lack organelles like the nucleus, which creates even more room for hemoglobin proteins which carry oxygen. While red blood cells are fantastic for gas exchange, the fact that they don't have any organelles means that they only live for about 120 days. So red blood cells are always being regenerated in the bone marrow. The thin white middle layer just above the erythrocytes is called the buffy coat, and it contains platelets and leukocytes, or white blood cells. This layer generally accounts for less than 1% of the volume of whole blood, with most of the volume being taken up by the leukocytes. Now, platelets are small pieces that split off of larger cells called megakaryocytes in the bone marrow. The main role of platelets is to clump together and form a plug that helps seal off a damaged blood vessel and prevent blood loss. And then there are the leukocytes, which are the only complete cells in blood, meaning they have all the usual organelles. There are lots of different types of leukocytes, and they all help to ward off pathogens like bacteria and viruses, as well as destroy cancerous cells and neutralize toxins. Some leukocytes are called granulocytes, because they contain tiny sacs called granules that are filled with inflammatory molecules. Neutrophils are the most common granulocyte, making up about 60% of the leukocytes, and these are usually the first to respond to an infection. Other granulocytes include eosinophils and basophils, but these only make up about 2 to 5% of leukocytes. Eosinophils are largely responsible for fighting off parasitic infections, while basophils are key in allergic reactions. Leukocytes that don't have granules include lymphocytes and monocytes. Lymphocytes include B cells, T cells, and natural killer cells, and make up about 35% of leukocytes. Lymphocytes are responsible for the adaptive immune response, which includes antibody production and is what allows our immune system to have memory, so that we can effectively respond to pathogens that have caused infections in the past. Lastly, there are the monocytes, which make up about 5% of the leukocytes, and these cells help gobble up bacteria or other pathogens via phagocytosis. Unlike the other blood components, leukocytes have the ability to leave the blood and enter the tissues using a process called diapedesis, which is where they slip in between endothelial cells that line the blood vessels. In a way, leukocytes are like a mobile army, utilizing the blood as a highway to get to different areas of the body. Now, the top layer contains plasma, which makes up approximately 55% of whole blood and is acellular, meaning it has no cells. About 90% of the plasma is water, and the rest is composed of proteins, electrolytes, and dissolved gases. The most abundant protein found in blood is albumin. Albumin is made in the liver, and it helps maintain oncotic pressure, which is the force that helps to keep water in the bloodstream. In addition, albumin is a transport protein. It shuttles fatty acids, calcium, lipid-soluble hormones, and even some medications around the body. Another group of plasma proteins are globulins, which include gamma globulins, which are antibodies that stick to pathogens and label them for destruction, as well as alpha and beta globulins, which transport fats, metal ions, and fat-soluble vitamins. Fibrinogen is another abundant plasma protein, and it's involved in clot formation for damaged vessels. Fibrinogen helps platelets attach to one another to form the initial platelet plug, and then other clotting factor proteins that are also in the plasma come along to stabilize the clot. All of these clotting factor proteins, including fibrinogen, can be removed from a sample of plasma, and when that happens, what remains is called serum. The electrolytes that are found in the plasma include mainly sodium, potassium, calcium, bicarbonate, and chloride. These electrolytes play vital roles in maintaining normal acid-base physiology in the blood, and help regulate blood osmolarity, which is the overall concentration of the blood. Other solutes in the plasma include hormones, nutrients like glucose, and respiratory gases like oxygen and carbon dioxide that are dissolved in the blood. Alright, as a quick recap. The blood is made up of about 45% erythrocytes, 55% plasma, which is mostly water but also contains proteins like albumin, electrolytes, and dissolved gases, and less than 1% is made up of platelets and leukocytes. Platelet plug formation, also called primary hemostasis, is the first of two steps needed for hemostasis. Hemostasis is how the body prevents blood loss when a blood vessel is injured or broken. Without hemostasis, even a minor injury would be life-threatening. Imagine dying from a nosebleed. During primary hemostasis, platelets clump up together and form a plug around the site of injury. Then in the second stage, called secondary hemostasis, the platelet plug is reinforced by a protein mesh made of fibrin. You can think of it like a brick wall where the platelets make up the bricks and the fibrin makes up the mortar that goes between the bricks. So going back to primary hemostasis, which is the clumping up of platelets, this step can be further divided into 5 more steps: endothelial injury, exposure, adhesion, activation, and aggregation. Let's imagine that you accidentally slice a tiny artery in your finger while cutting fruit. Ouch! When this happens, the knife cuts several layers of the artery. The innermost layer of the artery is the endothelium, and it's made of endothelial cells. Just outside of this layer are several layers of smooth muscle cells, which controls the size of the lumen, or the inner diameter of the vessel, by contraction and relaxation. Outside of the smooth muscle, there's a layer of protein, specifically elastic fibers that give the blood vessel the ability to expand and contract. Outside of the elastic fibers, there's connective tissue made up of collagen, which is the major structural protein in humans. This fibrous layer protects the vessel and anchors it to the surrounding tissues. So the first thing that happens when the knife cuts your finger is endothelial injury. When that happens, nerves that are attached to endothelial cells and the smooth muscle cells detect the injury and trigger a reflexive contraction of the smooth muscles near the injury site, called vascular spasm. This makes the vessel more narrow, to reduce blood flow and ultimately decrease blood loss through the damaged artery. Now, endothelial cells normally secrete nitric oxide and prostaglandins into the blood, which cause nearby smooth muscles to relax. When there's endothelial injury, secretion of nitric oxide and prostaglandins decreases, and the endothelial cells secrete a protein called endothelin instead, which causes the smooth muscles to contract. The second step is exposure, and that's when damage to the endothelial cells exposes the collagen that's below them. And damaged endothelial cells release a protein called von Willebrand's factor that binds to this exposed collagen. The third step is adhesion. Platelets, which are small fragments of larger cells called megakaryocytes, continuously circulate in the blood. When endothelial cells are damaged, platelets come in contact with the von Willebrand factor bound to collagen. And platelets have a surface protein called GP1b that allows them to bind to the von Willebrand factor proteins. The fourth step is activation. When platelets bind to von Willebrand factor via GP1b, the platelet gets activated, which means that it does a few things. First, the platelet changes its shape, and its membrane forms tentacle-like arms allowing it to grab onto other platelets. Second, platelets release more von Willebrand factor, as well as serotonin, which is a tiny molecule that attracts more platelets to the area, and calcium, which is useful in secondary hemostasis. Platelets also release adenosine diphosphate, or ADP, and thromboxane A2. These two molecules are secreted into the blood and activate other platelets that haven't bound to von Willebrand factor. So as more and more platelets bind, there's a snowball effect, and soon a ton of platelets are activated. This is a positive feedback loop. Now, we don't want all of our platelets to be used up just because we got a tiny cut on our finger. So the positive feedback loop is limited to the injury site. The way that works is that prostaglandin and nitric oxide secreted by undamaged endothelial cells binds to platelets and keeps them from getting activated. So we have this tug of war between prostaglandin and nitric oxide, which are platelet inhibitors on one side, and ADP and thromboxane A2, which are platelet activators on the other side. Finally, when ADP and thromboxane A2 binds to platelets, they express a new surface protein called GP2B/3A. Kind of like unlocking a new level in a game. Once GP2B3A is expressed on the surface of the platelets, they're considered fully activated. The fifth and final step is aggregation. Now, the ADP in thromboxane causes platelets to stick to collagen and cause free-floating platelets to express GP2B3A. GP2B3A binds to fibrinogen, which is a circulating blood protein that acts like a little pair of handcuffs that links two platelets together. Now, each platelet has multiple GP2B3A receptors. As a result, platelets on the collagen can attach to fibrinogen, and that same fibrinogen can be bound by platelets floating in the blood. This allows platelets to rapidly aggregate at the site of injury, and form a platelet plug that can help stop the bleeding. During secondary hemostasis, fibrinogen gets cleaved into fibrin, and fibrin forms a protein mesh, kind of like a giant net that covers the platelet plug and keeps it together. Alright, as a quick recap. Primary hemostasis is the first step of hemostasis, and is the process where a platelet plug forms to prevent further loss of blood from a damaged vessel. Primary hemostasis is divided into 5 distinct stages: endothelial injury, exposure, adhesion, activation, and aggregation.
Form a plug at the site of an injured blood vessel. While these platelets are aggregating, coagulation, or secondary hemostasis, begins. This is where numerous enzymes that are always floating around in the blood, called clotting factors, get proteolytically activated, meaning that activation happens when a small piece is chopped off, a bit like pulling the pin out of a grenade. These factors activate one another, eventually leading to the activation of fibrin, or factor 1A. That results in a fibrin mesh which forms around the platelet plug to reinforce it and hold it together. Without primary and secondary hemostasis, our body would suffer massive blood loss from even the most minor injuries. Imagine losing all of your blood volume from something as simple as a pinprick. Now let's get down into the nitty-gritty of secondary hemostasis. The process of forming the fibrin mesh begins via 2 pathways, the extrinsic and intrinsic pathways. The intrinsic pathway is called intrinsic because all of the factors required to activate it are intrinsic, or found within the blood. Conversely, the extrinsic pathway is called extrinsic because it's activated by tissue factor found extrinsically, or outside of the blood. Both pathways can become activated independently and ultimately culminate in the activation of factor 10, which then proceeds to activate the rest of the coagulation cascade via the common pathway. So let's start with the extrinsic pathway. It starts when trauma damages the blood vessel and exposes the cells under the endothelial layer, something like smooth muscle cells, which have tissue factor or factor 3 in their membrane. Now, it turns out that there's an enzyme called factor 7 floating around in the blood, and some of it is active, meaning that it's already set to proteolytically cleave other proteins. When it's in that state, it's called active factor 7, or 7A, A meaning active. Factor 7A binds to a tissue factor and a calcium ion, which get released by nearby activated platelets, and it forms a 7A-TF complex on the surface of the smooth muscle cell. Both the calcium ions and tissue factors are cofactors, meaning that they need to bind to the enzyme factor 7A to allow it to really get going. This complex then cleaves clotting factor 10, yielding the active form, called factor 10A. Once factor 10A is generated via the extrinsic pathway, it cleaves factor 5 into 5A, and then it uses factor 5A and a calcium ion as the cofactors to form the prothrombinase complex, which activates prothrombin, or factor 2, into just plain old thrombin, or factor 2A. In fact, each prothrombinase complex can activate thousands of thrombin, so there's an enormous amplification that occurs at this step. Thrombin uses calcium ions as a cofactor, and it has a number of procoagulative effects. First, thrombin binds to receptors on platelets, causing them to activate. Activated platelets change their shape to form tentacle-like arms that allow them to stick to other platelets. Second, thrombin activates three cofactors: factor 5, which is used in the common pathway, factor 8, and Fabin, which proteolytically cleaves fibrinogen, or factor 1, into factor 1A, also known as fibrin. Third, the effect of thrombin on fibrinogen plays a big role. See, fibrinogen is soluble in blood, but fibrin isn't, so fibrin precipitates out of the plasma and forms long protein chains that are like ropes. The fibrin tethers platelets to one another, holding the platelet plug together. And fourth, thrombin proteolytically cleaves stabilizing factor, or factor 13, into factor 13A. Factor 13A combines with a calcium ion cofactor to form crosslinks between the fibrin chains, further reinforcing the fibrin mesh. This is where the intrinsic pathway comes into play. The intrinsic pathway starts when a circulating factor 12 comes into contact with negatively charged phosphates on the membrane of activated platelets or subendothelial collagen exposed by trauma. Then it undergoes conformational change to get activated into factor 12A. Factor 12A proteolytically cleaves factor 11 into factor 11A, which combines with a calcium ion to proteolytically cleave factor 9 into 9A. Recall that thrombin also works at this step to activate factor 9 into 9A. Finally, 9A forms a complex with a calcium ion and factor 8A, and this complex proteolytically cleaves factor 10 into 10A. It's important to note that factor 8 degrades rapidly in the blood if it's not bound to a protein called von Willebrand's factor, which is released by activated platelets in endothelial cells during primary hemostasis. Now, there are two blood tests used to test coagulation. The prothrombin time, or PT, tests whether the extrinsic pathway is working. You can think playing tennis, which is done outdoors. Also, there's the activated partial thromboplastin time, or APTT, or just PTT, which tests whether the intrinsic pathway is working. You can think playing table tennis, which is done indoors. Now, to remember the various factors in both pathways, let's use a mnemonic. Let's start with Agent 007, representing Factor 7, who's sneezing into a tissue, for tissue factor, because he's been playing tennis, for PT, outside, for extrinsic pathway. Agent 007 then simply hops on the common train, for common pathway. Meanwhile, we have a farmer carrying 12 eggs, for Factor 12, on his way back from playing table tennis, for PTT, indoors, for intrinsic pathway. He clumsily drops an egg, so now there's only 11, for factor 11. Suddenly, a mean alley cat with 9 lives, for factor 9, jumps in his way. The farmer kicks his boot off at the cat, which hits it, and for the purpose of the mnemonic, the cat dies, so now that it only has 8 lives, for factor 8. The farmer then also hops on the common train. The train carriage has 10 seats available, for factor 10, but 5 of them are occupied, for factor 5. Some more people sit down, and soon there are only 2 seats left, for factor 2, and then just 1, for factor 1. Agent 007 and the farmer look at each other, and then scramble over to grab it. Who do you think got the seat? Alright, as a quick recap. Secondary hemostasis involves the formation of a fibrin mesh via the coagulation cascade. Coagulation starts when collagen and activated platelets activate the intrinsic pathway by activating factor 12, which activates factor 11, which activates factor 9, which activates factor 10. Factor 10 starts the common pathway where it activates factor 2, which activates factor 1 that builds the fibrin mesh. When factor 2 gets activated, it also activates 4 other factors, 5, 8, 9, and 13. Factor 5 gets activated and acts as a cofactor for 10. Factor 8 acts as a cofactor for factor 9, and factor 13 helps factor 1, or fibrin, form crosslinks. In the extrinsic pathway, exposed tissue factor activates factor 7, which activates factor 10 and starts the common pathway. Factors 2, 7, 8, 9, 10, 11, and 13 need calcium as a cofactor in order to function. Vitamin K helps to regulate the process of blood coagulation by assisting in the conversion of certain coagulation factors into their mature forms. Without vitamin K, our bodies would be unable to control clot formation. Imagine being unable to form blood clots effectively. That would mean we would lose all of our blood volume from something as simple as a pinprick.
Ow!
To prevent this extreme scenario, vitamin K must be ingested, metabolized, and utilized to create mature coagulation factors. Now, to understand the regulation of clot formation, we first need to talk briefly about hemostasis, in which hemo refers to the blood and stasis means to halt or stop. Hemostasis is divided into 2 phases, primary and secondary hemostasis. Primary hemostasis involves the formation of a platelet plug around the site of an injured blood vessel, and secondary hemostasis reinforces that platelet plug with the creation of a protein mesh called fibrin. To get to fibrin, a set of coagulation factors, each of which are enzymes, need to be activated. These enzymes are activated via a process called proteolysis, which is where a portion of the protein is clipped off. In total, there are 12 coagulation factors numbered factors 1 to 12, but there's no factor 6. Most of these factors are produced by liver cells, and it turns out that producing coagulation factors 2, 7, 9, and 10 requires an enzyme that uses vitamin K. Vitamin K is found in abundance in green leafy foods, things like spinach, kale, and chard. It's a fat-soluble vitamin along with vitamins A, D, and E, meaning that it can be stored in fat cells instead of being excreted by the kidneys. Vitamin K is also synthesized by bacteria in our gastrointestinal tract as a byproduct of their metabolism, which further contributes to overall intake. Now, when vitamin K is mobilized from fat cells or the digestive tract to the liver, it's in its dietary form, and it's called vitamin K quinone. An enzyme called quinone reductase takes electrons from NADPH and donates them to vitamin K quinone, converting it into the reduced form, which is called vitamin K hydroquinone. Then, vitamin K hydroquinone acts as a cofactor by donating its electrons to an enzyme called gamma-glutamyl carboxylase, which converts the non-functional forms of coagulation factors 2, 7, 9, and 10 into their functional forms. Gamma-glutamyl carboxylase adds a group, a chemical group made up of 1 carbon, 2 hydrogens, and 1 oxygen onto the end of glutamic acid residues on the proteins. After the carboxylation step, vitamin K is in an oxidized form where it can accept electrons, and it's called vitamin K epoxide. Vitamin K epoxide gets converted back into vitamin K quinone by another enzyme called epoxide reductase, which donates electrons to vitamin K epoxide using a thiol group. In this fashion, a single molecule of vitamin K can be reused many times. As it turns out, the drug warfarin, which was first used as a rat poison, blocks the function of this enzyme, which blocks vitamin K from getting recycled, and as a result, factors 2, 7, 9, and 10 don't get activated. Now, let's take a closer look at the coagulation cascade to see where these coagulation factors play their respective roles. The coagulation cascade begins via 2 pathways, the extrinsic and intrinsic pathways. The intrinsic pathway starts when circulating factor 12 comes into contact with the surface of activated platelets or collagen. Activated factor 12 then activates factor 11, which activates factor 9, which activates factor 10. Factor 10 starts the common pathway where it activates factor 2, which activates factor 1 that builds the fibrin mesh. When factor 2 gets activated, it also activates 4 other factors: 5, 8, 9, and 13. Factor 5 gets activated and acts as a cofactor for 10. Factor 8 acts as a cofactor for factor 9, and factor 13 helps factor 1, or fibrin, form crosslinks. In the extrinsic pathway, exposed tissue factor activates factor 7, which activates factor 10 and starts the common pathway. So, without vitamin K, the loss of factor 7 means that the extrinsic pathway won't function. And without factor 9, the intrinsic pathway won't function. And without factor 10 and 2, the common pathway won't function. Since these factors are utilized in all 3 pathways, it's easy to see how clot formation does not function properly unless our body properly utilizes vitamin K. Alright, as a quick recap. Vitamin K plays an essential role in clot formation because it's used in synthesis of clotting factors 2, 7, 9, and 10. In order to be useful, vitamin K undergoes a series of oxidation and reduction reactions called the vitamin K cycle. An important enemy of this system is the drug warfarin, which inhibits epoxide reductase to prevent maturation of these factors.
In hemostasis, hemo refers to blood and stasis means stop, so hemostasis is the process where blood flow is stopped after there's damage to a blood vessel. Primary hemostasis involves the formation of a platelet plug at the site of an injured blood vessel, and secondary hemostasis involves the coagulation cascade, which is where a protein net called a fibrin mesh forms over the platelet plug to reinforce it, forming a blood clot. Now, anticoagulation occurs during primary and secondary hemostasis and helps to regulate clot formation, whereas clot retraction and fibrinolysis occur after primary and secondary hemostasis are complete and help a clot contract and degrade. Anticoagulation prevents clots from growing too large and blocking blood flow to tissues supplied by the vessel. It also prevents clots from getting so big that small parts of the growing clot break off and form emboli. Depending on the location of the primary blood clot, these emboli may then cause a disruption in blood flow to organs like the heart and brain. Now, the most important part of this clot regulation is when a coagulation factor called thrombin is produced. Thrombin, or factor 2, is a very important clotting factor because it has multiple procoagulative functions. Think of thrombin as an accelerator on a car, the pedal that takes the secondary hemostasis from 20 miles per hour to 100 miles per hour. First, thrombin binds to receptors on platelets, causing them to activate. Activated platelets then change their shape to form tentacle-like arms that allow them to stick to other platelets. Second, thrombin activates two cofactors, factor 5, used in the common pathway, and factor 8, used in the intrinsic pathway. Third, thrombin proteolytically cleaves fibrinogen, or factor 1, into fibrin, or factor 1a, which binds other fibrin proteins to form a fibrin mesh. And finally, thrombin proteolytically cleaves stabilizing factor, or factor 8, into factor 8a. Factor 8a combines with a calcium ion cofactor to form crosslinks between the fibrin chains, further reinforcing the fibrin mesh. Since thrombin has so many jobs, it makes sense that it has the main target of two proteins that help with anticoagulation, protein C and antithrombin 3. Protein C is a circulating plasma protein produced in the liver, along with a cofactor called protein S. Now both protein C and S interact with a protein called thrombomodulin, which is on the surface of intact endothelial cells which line our blood vessels. Now let's go back to an existing clot. When there's a lot of thrombin around a damaged blood vessel, excess thrombin binds to thrombomodulin and it can no longer participate in the coagulation cascade. So in a sense, the undamaged cells help to ensure that the coagulation process is limited to the injury site. And furthermore, the thrombin-thrombomodulin complex binds to and activates protein C and S. The whole thing forms a complex that includes protein C, protein S, and thrombin-thrombomodulin. This protein complex proteolytically cleaves and inactivates active factor 5, a cofactor for factor 10 in the common pathway, and 8, a cofactor for factor 9 in the intrinsic pathway. By inhibiting both the intrinsic and common pathway, coagulation slows down dramatically. Now, a second anticoagulant is antithrombin 3, sometimes just called antithrombin. Antithrombin is a protein made by the liver and released into the blood, and it binds to both thrombin and factor 10, both of which are in the common pathway. Excess thrombin can bind to antithrombin, similar to how it binds to thrombomodulin, and become unavailable. Antithrombin also binds to excess factor 10, which is a pivotal coagulation protein that converts prothrombin into thrombin. Antithrombin also inhibits factors 7, 9, 11 and 12, although with much less affinity. Antithrombin is also the target of an effective medication called heparin. Heparin binds to antithrombin and increases its affinity for its target proteins, thus increasing its anticoagulant effects. So when individuals are given heparin, the balance between coagulation and anticoagulation tips in favour of anticoagulation. Now, there are also some factors that prevent platelets from adhering to one another or to endothelial cells during primary hemostasis. Healthy, undamaged endothelial cells release nitric oxide and prostacyclin. These tiny molecules make activated platelets produce less thromboxane A2, which is a molecule that active platelets secrete to activate surrounding platelets. Specifically, thromboxane A2 makes platelets express GP2B3A proteins on their membrane, and GP2B3A proteins bind to fibrinogen, which ultimately makes platelets stick to one another. Now, within an hour or so after primary and secondary hemostasis creates a blood clot, platelets begin the process of clot retraction. Activated platelets have proteins within them called actin and myosin, the same proteins found in muscle cells. These proteins are arranged in bundles called lamellipodia, and from the outside of the platelet, they look a bit like fingers poking through the platelet membrane. A little bit creepy. Anyway, these lamellipodia are really important because they increase the surface area of the platelet, and that makes it easier for them to bind to the fibrin mesh as well as the endothelial lining of the blood vessel. Now, platelets that are part of a clot typically have a surface protein called integrin alpha-2b beta-3, and when these receptors bind to fibrin in the presence of thrombin, it activates actin and myosin. Actin and myosin slide along one another and it causes lamellipodia to contract, and since the cell surface integrins are both bound to fibrin, it pulls in on the fibrin mesh. The fibrin mesh tightens in on all of the platelets in the clot, making the whole thing more rigid and contracted down. When the clot contracts, it brings the edges of the wound closer together. When there's an open wound, the clot retraction pulls the two wound edges closer together, and the endothelial cells can divide and replace the lost tissue. If the gap is too wide, it can get filled in with collagen, but this process takes longer and typically results in scarring. Now, approximately 2 days after an injury, the blood clot is no longer needed since substantial healing has taken place. At that point, it's time for the body to dissolve the blood clot through a process called fibrinolysis, which is the gradual degradation of the fibrin mesh. To do this, a circulating protein produced by the liver called plasminogen gets converted by an enzyme called tissue plasminogen activator, or TPA, into its active form called plasmin. Normally, healthy endothelial cells release only tiny amounts of TPA, but when they're exposed to coagulation factors, mainly factor 10 and thrombin, they start making lots of TPA. But it's important that plasmin activity doesn't get out of hand either. So the endothelial cells also release plasminogen activator inhibitor 1 and antiplasmin, which are proteins that bind and sequester plasminogen and plasmin respectively. It's all about always reaching that zen balance of coagulation and anticoagulation. Now, during an injury, more and more coagulation factors get activated, and also more and more TPA gets released by endothelial cells, converting plasminogen into plasmin. Plasmin then acts as a protease, literally cutting fibrin into smaller pieces, allowing the trapped red blood cells and platelets to float away, letting the clot dissolve. Although TPA is the main activator of plasmin, a few other proteins activate plasmin as well, including coagulation factors 9A, 7A, kallikrein and protein C. TPA is also used clinically. It's sometimes called a clot buster because it's used to dissolve blood clots that form in the lungs or in the brain. Okay, so as a quick recap, anticoagulation with protein C and S and antithrombin 3 prevents clots from getting too big during primary and secondary hemostasis. Within an hour or so after primary and secondary hemostasis have created a blood clot, clot retraction begins to stabilize the clot and starts pulling together the wound edges. Finally, there's the process of fibrinolysis, which is dependent upon the TPA, which converts plasminogen into plasmin, an enzyme that cleaves fibrin and helps to dissolve the clot.